Every single cell in your body, all trillions of them, is like its own bustling city. It's constantly trying to bring in supplies and ship out waste. But here's the catch. How does it handle all that traffic when its borders are basically on lockdown? Well, today, we're going to break down the amazing security systems that literally keep your cells alive.
So let's really lean into that idea. Think of a cell as a fortress, right? It's got this big wall to protect all the precious stuff inside from the chaotic world outside. But a fortress that can't get food or water or even a message in or out, that's not a stronghold, that's a tomb. So how in the world does the cell solve this massive paradox?
The whole answer, it turns out, starts right there at the border. The problem, and, brilliantly, the solution, is all about that fortress wall. It's a structure we call the plasma membrane.
Okay, so what is this plasma membrane? Well, it's a lipid bilayer. Just picture two layers of fatty molecules, kind of like a microscopic oil slick, all packed together. This creates a barrier that's flexible but really, really effective at separating what's in the cell from what's out. It's an incredible protector, but man, is it picky about who gets a hall pass. And this right here is the central problem. The membrane is totally cool with small, uncharged stuff like oxygen and carbon dioxide just slipping right on through. But it's like a bouncer at a club for almost everything else the cell desperately needs to survive. We're talking charged ions like sodium and potassium, essential fuel like glucose, huge molecules like proteins. Nope, denied. So the wall is amazing for defense, but it's just awful for trade. The cell needs a totally different strategy.
All right, let's start with the simplest solution. For that very exclusive list of molecules that can get through, they do it by, well, just going with the flow. This is called simple diffusion. And really, it's just the universe's natural tendency to spread things out. Molecules will always move from a place where they're super crowded to a place where there's more room. It just happens. It's automatic. No energy required, no special helper needed, just pure physics doing its thing.
You can think of oxygen molecules like tiny little spies, just slipping right through the cracks in that fortress wall. So when you take a breath, there's suddenly a high concentration of oxygen in your lungs, and boom, it diffuses right into your blood cells. And at the exact same time, your cells have all this carbon dioxide waste built up, so it diffuses out. It's simple, it's elegant, and it's totally vital.
But here's the thing. Simple diffusion only works for a tiny VIP list of molecules. For absolutely everything else that's stuck at the border, the cell has built something incredible, a series of special, gated passageways. So this next method is called facilitated diffusion. Now the key here is that it's still passive. That means stuff is still moving from high to low concentration, and it still doesn't cost any energy. The big difference is that it needs a facilitator, a helper. And that helper is a protein channel that acts like a secure tunnel or a gate right through the membrane. And what's just wild is the sheer variety of these gates. I mean, cells have channels that pop open in response to chemical signals or physical force or even changes in electrical charge. Some even respond to light. They've even got these specialized water-only channels called aquaporins, just to help water move in and out faster. Each gate has its own very specific job.
To make it even clearer, just think of it like this. A ligand-gated channel. That's like a door that only opens with a specific keycard. A chemical has to physically bind to it. A mechanically gated channel is like a pressure plate on the floor. A physical force, like the sound waves hitting cells in your ear, literally pushes it open. And a voltage-gated channel is like an electric switch. It flips open when the electrical charge changes, which is basically how your nerves fire.
And how do we even know all this? Well, scientists developed this incredible Nobel Prize-winning method called the patch clamp technique. It lets them zoom in and measure the electrical current flowing through a single ion channel. And what they found was that these gates aren't like dimmer switches. They are strictly all or none. They are either 100% open or 100% shut, no in between. Okay, so everything we've talked about so far involves moving down a concentration gradient, from a crowded area to a less crowded one. But what happens if a cell needs to do the exact opposite? What if it needs to stockpile supplies, to hoard something, moving it from where there's very little to a place where there's already a ton? Well, for that, it has to work against the current. And this is where we finally meet active transport. And here is the single biggest difference. Unlike everything else, active transport moves things against their natural flow, from low concentration to high. And to fight against physics like that, you have to pay a price. It requires energy, which the cell pays for with a molecule called ATP. So just how much energy are we talking about here? Honestly, it's staggering. Get ready for this. Your average animal cell uses nearly 1/3 of its entire energy budget just to run one single kind of active transport pump. A third. That tells you just how unbelievably important this process is.
So just picture this happening millions of times a second. There's this famous pump called the sodium-potassium pump. First, it grabs 3 sodium ions from inside the cell. Then, ATP comes along and gives it a jolt of energy, forcing the pump to change its shape and spit the sodium out. Now that it has this new shape, it grabs 2 potassium ions from the outside, and then it snaps back to its original shape, releasing that potassium inside the cell. It's this constant, relentless, energy-guzzling cycle.
So why on earth spend 1/3 of all your energy on this one pump? Well, because this single machine is a master multitasker. It creates the negative electrical charge inside our cells that allows our nerves to send signals and our muscles to contract. It controls the water balance to keep cells from literally swelling up and bursting. And that huge sodium gradient it builds, that's used as a power source for a bunch of other transporters. It is, without a doubt, a cornerstone of animal life.
So we've seen this beautiful, complex system of gates and pumps all working together perfectly. But remember, this machinery is all built from proteins, which are encoded by our genes. So what happens when one of those genes has a mistake, and one of these tiny, critical gates just breaks?
The consequences, as you can imagine, can be just devastating. We now know that a whole host of inherited diseases are caused by nothing more than a faulty ion channel. A broken chloride channel can cause cystic fibrosis. A glitchy potassium channel can lead to a dangerous heart rhythm problem called long QT syndrome. And issues with sodium channels, they're linked to certain kinds of epilepsy and high blood pressure.
Let's just focus on cystic fibrosis for a second. The entire disease, all of its symptoms, comes down to one specific chloride channel that just doesn't work right. Because these chloride ions can't get out of the cell, the water balance gets all thrown off. This leads to this incredibly thick, sticky mucus building up in the lungs and other organs. It's a perfect, tragic example of how a single, tiny defect at the molecular level can cause a life-threatening cascade of problems throughout the entire body.
So just think about that for a second. Your body's survival, right now, depends on literally trillions of these tiny gates opening and closing, and these pumps working non-stop every single second of your life. These aren't just some abstract thing in a biology textbook. They are the true gatekeepers of life itself. And that leaves us with this really fascinating question for the future. As our understanding gets better and better, what happens when we learn to control them? To fix the ones that are broken, and maybe even build new ones from scratch? The possibilities? Well, they're nothing short of extraordinary.